Hi everyone, this little video is about the proteome. It's aimed at the level of detail for advanced higher biology. This is Unit 1, which is cells and protein. Key Area 2, this is just the first bit of Key Area 2. We are aiming to find out what the proteome is and look at why and when different genes exhibit different proteins. Okay, we're going to look at the types of things that's going to control what proteins are produced at different points in a cell's life, and also in different types of cells, because specialized cells will be exhibiting the proteins that are required for them to carry out their specific jobs. Before we look at the proteome, let's have a little reminder about the genome. Right, you should be fully aware that the genome is all the genetic material in a cell, including DNA and RNA. Genome, genetic material. Okay, so with that, when we look at the proteome, we're looking at the proteins. The proteome is the entire set of proteins that is or can be expressed by the genome. Okay, notice I said or can be. All the proteins are not going to be expressed all the time. Okay, so genome is genetic material, proteome is proteins. Have a little think. Which one is bigger, the genome or the proteome? We know the genome's huge. However, the proteome is even bigger. You'll know from higher biology that one gene can actually be transcribed and translated into a number of different proteins. Okay, so this means if our genome is, let's say, 20 to 25,000 genes here, then our proteome actually ends up being over 1 million proteins. Okay, if we just look here. And at higher biology, we looked at things like alternative splicing. We could also have alternative promoters, so different parts of the gene get turned on at different times. When we're actually transcribing the gene due to RNA splicing, which we'll go into a bit later, we actually end up including different bits that get pushed forward for the translation into the protein. And of course afterwards, which we'll also look into greater detail, after transcription, when we go to translation, there can actually be some post-translational modifications. We can be adding different things onto our proteins. So that will be one gene giving quite a variety of different proteins. So the proteome is larger than the genome. The number of possible proteins that can be produced from the genome is greater than the number of genes. Okay? And as we know, more than one protein can be produced from a single gene as a result of alternative RNA splicing, which is something we looked at higher. Just as a reminder, got this diagram here. Now remember, when we get to RNA splicing, what happens is our messenger RNA is split up into introns and exons, and all those introns are removed and all the exons are joined together, and it's only the exons that go forward for translation. Now alternative RNA splicing is when different sections of the messenger RNA are treated as introns and exons in one instance, but then in another instance, it could be different areas that are the introns and the exons. So what we've got here is we might have the same RNA here, there's our RNA, but in actual fact, we go forward to translation, we've got 3 different scenarios where we've got 3 different lots of exons. Okay, so that results in 3 different proteins. Now there are also non-coding RNA genes. So these will be genes that go forward and do not become proteins. They become RNA. Okay, you should hopefully remember the 3 types of RNA. We've got tRNA and rRNA, and of course mRNA, but mRNA has already been used, okay, because remember this is after transcription. So our non-coding RNA genes will code for things like tRNA and rRNA, and also RNA molecules that control the expression of other genes, okay? These are RNA molecules that will turn on or off different genes other than themselves. Another thing that can happen with protein expression, not only do you have some genes producing a number of different proteins, it's also protein expression changes over time and in different conditions. Okay, so when you are a baby, you do not have all your proteins being expressed. You've not got the same set of proteins being expressed in a baby as you do in an elderly man. Now have a look at this diagram here. Right, we're looking at the early embryo of an insect going through to the later embryo and then the larval stage. We're looking at the expression of the gap-krupel gene. Okay, in the early embryo, you can see here that there's only a small region of expression of this particular gene. Later in the embryo, later in development of the later embryo, you can see that there is more cells expressing that gene. And then further on in the larvae, these cells are still expressing the gene. So, early on in the embryo, we had a little expression, and then over time, it increased in expression. We can also have the situation of protein being expressed under different conditions. Factors that are going to affect the set of proteins being expressed by a cell might be the metabolic activity of the cell. Has it got a high metabolic rate? That's going to affect what proteins are being expressed. Okay. Is it under cellular stress? When there's cellular stress, there'll be certain hormones and chemical cues and signals that are given off. Okay. Gene expression will respond to this. It's going to respond to signaling molecules as well as chemical cues. And of course, if you've got a disease cell versus a healthy cell, there's going to be a different set of proteins that are being expressed there as well. So in summary, now this is a point where pupils will often take notes from the board. So if you want to, you can pause the video at this point and take down any notes that you like. Okay. So in summary, the proteome is the entire set of proteins expressed by a genome. It is larger than the number of genes that the genome has because more than one protein can be produced from a single gene as a result of alternative RNA splicing. Genes that do not code for proteins are called non-coding RNA genes, and they can be transcribed into tRNA, RNA, and other molecules that control the expression of other genes. The set of proteins expressed by a given cell can vary over time and in different conditions. Factors that will affect the set of proteins being expressed are the metabolic activity of the cell, cellular stress, response to any signaling molecules that might be present, and whether the cell is diseased or healthy. Hopefully that will have helped you with understanding a bit about the proteome. We were trying to define the term proteome, explain why it's larger than the genome, and be able to describe some of the factors that are going to affect the set of proteins that are being expressed by a cell at any one time.